Okay, Mia Robinson 517789, attempting presentation on ATP and the glycolysis through to ETC pathway. I am trying to get this slide in some kind of focus. You'll have to bear with me because it's a bit Blue Peter. Yes, so let's begin. Okay, Mia Robinson, student, no disclosures for the purpose of this presentation. We will be discussing ATP molecule, its energy potentials in all living things, and the pathways, focusing specifically on humans in this particular presentation, although ATP is used by all living things. Slightly different pathways, but essentially the same mechanisms. So what is ATP? Adenosine triphosphate, the universal currency of energy, because, as I mentioned, used by all living things, consists of an adenine base, a ribose sugar, and 3 phosphate groups. They can be represented by zeros or OHs, phosphohydrogen bonds. Depends how you want to represent it, or symbolically they can be represented by 3 circles, the pentagram for the ribose sugar and the adenine base. The phosphate groups are labelled alpha, beta, gamma through hydrolysis, which is hydro meaning water, lysis to split. The beta to gamma phosphate can be broken off, resulting in the formation of adenosine triphosphate, adenosine diphosphate, di being 2, and the release of 7.3 kilocalories of energy per molecule potential. This can then be broken down again into adenosine monophosphate, meaning 1 phosphate. There's also cyclic AMP, which is used in certain pathways. All of these can be recharged. Hydrolysis used to split them up. Phosphorylation, mainly by ATP synthase, discussed later in ETC, is what brings them back to full charge and back into the cycle to be used again. Okay, point of fact, at any one point, resting state, there's probably approximately 1.5 kilograms of ATP being used at any one time, according to sources. Okay, so glycolysis, where does this happen? Okay, so this glycolysis, glucose is brought into the cells, or molecules are brought into the cells through the receptors. This is the cell membrane here, so we go into the cell. The glucose is brought into the cell. We have our 6 carbon chain here. ATP comes along and it releases a inorganic phosphate, turns into ADP. So 1 times ATP is used to turn this into still a 6 carbon chain, but the organic phosphate has been added to the end here. Again, we go through the similar process where, again, another inorganic phosphate group is put on the end of the 6 carbon chain. Miss a few steps here, like fructokinase and a few other ases, and we come down to, I have represented with the 2 pathways because it's easier for me, the 2 3 carbon chains with the inorganic phosphate group attached at the end. We go for a few other phases here that I haven't discussed, where basically this phosphate group just moves around a little bit, and then the ADP comes along and it phosphorylises with this phosphate group, turns back into ATP. And again, the same thing happens over here. So we have 2 ATP used, but we have a net of 4 ATP, 2 ATP used, 2 ATP produced, but we also have some NAD3H as well. So we bring that back down to this chain. We've got this 3 carbon molecule, the phosphate's been removed. We come to pyruvate. Pyruvate and lactate, very similar molecules. If pyruvate is brought over to the mitochondria in the presence of oxygen transporters will help it cross the membrane into the mitochondria. This is an anaerobic process. This becomes an aerobic process. In anaerobic conditions, we've got decreased oxygen here, increased oxygen or oxygen good saturation. It will go through into the mitochondria. If we have a lack of oxygen, we're going to go back into this lactate cycle. Over lactate production, we're going to get lactic acid, can cause acidosis, can also be a feature in alcoholic liver syndrome, where ethanol and CO2 can build up, affecting the NAD, NADH plus concentrations, and ethylene. It can produce a small amount of ATP, but it's not optimal, and sustained reactions like this can be very, very damaging. So in a summary here, anaerobic is with oxygen. Pyruvate moves on to the link reaction into the mitochondria. In anaerobic conditions, without the O2, it's unable to move on, and it goes back into lactate. I mean, these cycles are completely reversible. Lactate can come pyruvate infinitum, and the lactate and fatty acids are a big build-up of fatty liver, as I'm sure I've mentioned. Apologies, not too well at the moment. So we move on to the link reaction, and this is converting pyruvate into acetyl-CoA. Pyruvate at this stage is decarboxylated, and that means basically the carbon atom is removed and also oxidized, by acetate. This is my lovely drawing here. If we had a slide, this would be the pyruvate dehydrogenase complex, which is an enzyme reaction that goes on about here that I'm not going to mention. So as we come into the link reaction, we're coming into the mitochondrial matrix space. We bring our free carbon pyruvate, which is brought in thanks to the oxygen reaction. The next stage is it decarboxylation. So there goes the carbon, there goes the oxygen, and we got waste of CO2. As a result of this, we get NAD. NAD becomes reduced, which is a weird terminology. It actually means you've added a hydrogen proton through oxidative phosphorylation. So going on as well. We come down to a 2 carbon atom, because obviously we decarboxylated one off. We then add some coenzyme A, or CoA as it's known, and in the pyruvate oxidation is final step linked glycolysis. And and acetyl-CoA. Basically, these are all ×2 because this reaction will be happening ×2. And that is in the mitochondria. The acetyl-CoA can go on to Krebs, and it can go on to some other functions as well. If I was at college, I would have had a slide of that one. No ATP is produced in the link reaction, but too much acetate in this reaction can be very harmful. Again, we're going back to the alcoholic situation here, where, other chemicals can be used in these pathways, but sustained use of is damaging. Okay, now we're going to go all the way over to the citric acid cycle, otherwise known as the Krebs cycle. Okay, now this is the mitochondria. We're focusing here in the mitochondrial membrane. So we have our 2 carbon atom acetyl-CoA has come down here. We have a CoA that comes off. So, here we go. This is a fun one. So we come down here. We've got a 6. We go into a 6 carbon chain, because this 4 carbon is actually going to get reproduced. A soxyl acetate is going to get added to this acetyl-CoA. We're going to go around to a 6 carbon substrate of citrate. We're going to reduce some NADH in this process to get to alpha ketoglutarate. After alpha ketoglutarate, we do have succinyl and succinate. At this point, we get a carboxylation reaction, so we're taking away a carbon atom and oxygen. NADH is then reduced again, so we're producing some more positive hydrogen protons there. So we have another 5 molecule carbon, succinate substrate. Again, we are decarboxylating. Off comes the carbon. We've got an NAD being reduced. We've got ADP plus an organic phosphate being turned into ATP. We've got an FAD being reduced to FADH, NAD. Again, there are some other substrates in these processes. I haven't gone all into them. There's a very lovely but rude acronymic, whatever you want to call it, to remember them, but I shan't go into that. Anyway, we go through these different steps, and we've got fumarate. Eventually we come to oxytalate, where ATP ATP is then phosphorylized again into ATP, and acetyl-CoA comes back in, and we start this again, hence the name. The cycle. Decarboxylation again, it's CO2 removed, taking the carbon molecule away. 38 ATP are theoretically produced through all cycles, and that is including the figures for the FADHs and ADHs. At this point, I'd like to mention that GDP, guanosine triphosphate, can be used in the same way, very similar makeup as adenosine triphosphate can be used, is used in some pathways, don't need to mention it here, but for the purpose of this little portion, we have outcome here of we've got 2 ATPs, we've got AA and ADHs, which add up to 3 ATPs each, we've got 2 FADHs, 2 ATP, and we've got the cycle going on a few times here, so we've got a net potential of 38 ATP. There are many more steps in this pathway, but I'm not going to cover all of them at this level, as I have been told. Right, okay. So to the electron transport chain, and this is really where all the magic happens, in my opinion. And that's not what this is about. It's about the science. So, okay, well here we have the outer mitochondrial membrane, the inner mitochondrial space. Here is the inner mitochondrial membrane. We're going on to the the inner mitochondrial membrane. Oh, side note, I may not have mentioned that the glycolysis cycle happens in the cytosol, cytoplasm of the cell prior to entering the mitochondria. This all happens within the mitochondria. It starts from the Krebs coming up through the matrix into the intermembranal space via these particular channels. They do have some names, all numbers 1, 2 and 3. There is actually a 4th pathway into there, but I don't think we need to get into this at this stage. So, oxidative phosphorylation or chemiosis, osmosis. Anyway, we're going to go through step 1. Hydrogen is released. Hydrogen protons, positive hydrogen protons, are released for the reduced NADs, FADHs. They are oxidized and then split into the positive hydrogen protons and the negative electrons. The electrons are going to come up here, they're going to go into these inter-channel spaces, and they're going to make their way across. Now they come down to the 3rd complex and they come out, they lose potential energy each time they cross one of these spaces. The hydrogen protons are going to make their way from this low concentration of gradient up here to the intermembranal space, where they're going to build up into a high concentration. Yes, it's the negative electron energy that actually helps propel the positive hydrogen protons through the intermembrane space. Okay, yeah, so we've created an electrochemical gradient here. We've got a low gradient down here, a high gradient down here. So the next thing that happens is these hydrogen atoms got to find a way back down into the low concentration gradient. The way they do that is this wonderful molecule called ATP synthase. I cannot show you the wonderful 10-second video of this process occurring with this beautiful molecule in action. Basically looks like a bit of a turbine, and the process of ATP production through the chemiosmosis, which I can never say, the hydrogen protons comes down here, and there's basically like a turbine that spins here. ADP plus an organic phosphate is brought in, the hydrogen and proton fuses these two together, rephosphorylizes, turns this back into ATP. And we've got these electrons that have come down here into this pathway. We're mixing, we've got some free floating oxygens, some hydrogen atoms around, and this slowly forms water, which is again used in hydrolysis reactions. Chemiostatic theory, which is the movements of ions across the semi-permeable membrane down an electrochemical gradient. I think we've already covered that kind of stuff. In the mitochondrial matrix, the hydrogen electron atoms and O2 from the blood combine to form H2O, and that's that one there that I was talking about. That O2 there is coming from the bloodstream, forms a H2O, which is needed for other body processes to go back into these chains. When it goes down these chains, there are certain molecules that are electron donators and are electron receptors, and things can be passed down the chain via different molecules. These electrons can jump around, but O2 in this particular pathway is a final electron acceptor. It's a universal. That's where your electrons are going to stop, especially they've been pulled down here. Hope that made some form of sense. Anyway, really hard to do it from home with my Blue Peter squares. Please, Tony, if I miss something, give me a shout and I will run back over it. Thank you very much. Meow.